Welcome back. This mini lecture will be about deep sea adaptations as well as deep sea environments. Let's start off with deep sea adaptations. So, picture yourself as a deep sea animal. How will you deal with no light, immense pressure, and near freezing temperatures? Pause the video, take about 3 minutes to brainstorm, and jot down some ideas as to what sort of adaptations you may evolve to have. Mind you, there are no right or wrong answers. Okay, pause the video now. Let's start off with adaptations for the darkness. With my all-time favorite thing, actually, I have lots of all-time favorite things, but one of my all-time favorite things, bioluminescence. Bio means life. Luminate, luminescence means light. So there are two types of bioluminescence in the deep sea. Most bioluminescence in the deep sea is a form of bacterial symbiosis. Symbiosis. So these are organisms that live together, symbiotes. These are not free-living dinoflagellates. The bacteria that cause the bioluminescence is cultured by the host, aka the fish or the crab or the plankton, whatever harnesses that bioluminescing bacteria is the host. It's still speculated as to how the actual bacteria got there in the first place. The fish are most likely born with it. However, scientists still don't know. All right. Number 2, specialized mitochondrial cells in the organism called photocytes. The animal produces light itself using nerve/electrical impulses that triggers the light-producing enzyme luciferase. Like I said earlier, a photophore is a light-producing organelle. An organelle is a tiny organ. We all have organs in our bodies, like our heart and our lungs and our kidneys. An organelle is just a tiny organ. L means tiny. So, photophore is a light-producing organelle that contains either the symbiotic bacteria or photocytes. On the right side of the screen, you can see these circular little dots on this myctophid right here, this fish. Those are the photophores. In the middle picture is a squid. You can see those light-producing organelles, photophores. And then on the left, the quintessential deep sea animal, an anglerfish, has one giant organelle on its esca right there. One giant photophore. Okay, moving forward. So this anglerfish, like I just said, has one gigantic photophore. You can see it right here in this image, producing light on a lure. This whole structure right here is called an esca, E-S-C-A. That's called an esca, with the photophore on the very tip. And the purpose of this, its evolutionary adaptation, is to bait or lure in unsuspecting prey, and it's used for hunting. Also, bonus fun fact, the esca is actually a modified fin ray. Evolution is so cool. All right, now for hatchet fish. Family Sternoptichidae. I'm not sure if I said that right. This is probably the very most cryptic fish. It's great with camouflaging because it has silver scales that act like mirrors. So these silver scales mirror the water column so that fish and hunters around this fish can't see the fish because it's a reflection of what is around it. On top of that, it has photophores, bioluminescing photophores on the underside of the fish, which is like counter shading, but one step further. So predators below the fish looking up see the lighter underside. It blends in with the lighter upper pelagic as the predator is looking up in the water column. And then oppositely, it does not have these photophores on its dorsal side, on the top side of its body. So when predators are above the fish looking down, it is also very cryptic. Okay, blue lanternfish within the family Myctophidae. So this also has counter illumination, which is like one step further than counter shading. And you can see these little dots on the underside of these myctophid fish. Those are the photophores that exhibit counter illumination. Here is a headlight fish, which is also a species within that same group of Myctophidae. There are two giant photophores on their forehead, on their face, that resemble headlights. And that's why they're called the headlight fish. This is used for hunting. Just as you shine your high beams when you're driving late at night and you can't see very well, these fish have evolved to have essentially two headlights to see their prey ahead of them as they're hunting. You can see these two photophores in action in this picture right here. All right, let's take a little pit stop in my presentation for myctophids, aka the lanternfish. So this group of fish is the most ubiquitous, abundant, they're everywhere, the most ubiquitous fish in the world. There are 33 genera and 246 species. They are everywhere in the twilight zone and the midnight zone. They're so cool. So here you can see all the different species. The bottom right is a headlight fish. Here is a Mexican myctophid. And then right here is a blue lantern fish. And those are the three species that I'm able to ID out of this gorgeous group. All right, moving forward. Okay, so on one of my deep sea cruises, I was lucky enough to pull up some fresh myctophids, and their photophores were still working. And I learned that their photophores were still producing light because I walked into this walk-in freezer, which was the only place that I could find that I was able to turn the lights off. And I knew that this room would become pitch black if I did so. So I walked in this walk-in freezer, myctophid in tow, in my hands, and I closed the door, turned out the lights, and let my eyes adjust for a little bit. And then I stimulated the photophore with my finger, and I saw light. So I was able to actually see in real time, in real life, a photophore on a myctophid, and it was one of the coolest things I've ever seen in my whole life. It was worth walking into this walk-in freezer and closing the door and turning out the lights. It definitely was a little bit creepy. And there was a panic button somewhere in there, but it was dark, so I couldn't see it. But I made it out alive, and I saw a myctophid photophore bioluminesce. Pretty darn cool. Okay. So red light is the very first wavelength of light to dissipate in the water column. This means that red light cannot make it down to the depths of the oceans. The first one to lose energy in the upper pelagic and not make it down to the deep sea. Blue light, however, is able to make it down to the deep sea. So, since red light does not make it down to the deep sea, a lot of deep sea animals have evolved to be red, to be cryptic, and to be camouflaged in the deep sea. So when these organisms are red, there's no red light down there, so their predators cannot see them. And one of my favorite aspects about this is that all of these species are different. They're different. This anglerfish is different from this jellyfish, which is different from this crab. And all of these species have convergently evolved to be red. They're not related, they're unrelated, yet they have all undergone the same evolutionary processes to become red, which is called convergent evolution. Just like how a shark and a dolphin have the same torpedo fusiform body shape that's very hydrodynamic, that is another example of convergent evolution. They are unrelated, yet they look very similar. They have similar characteristics because of the same environmental pushes. Okay, so some organisms in the deep sea have evolved to have huge eyes to try and absorb as many photons as they can in the deep sea because photons are scarce, right? There's not very much light in the deep sea. So these animals have evolved to have giant eyes with giant pupils to try and find as many photons as they can. Okay, and then also on the opposite side, a lot of organisms have evolved to have no eyes. No eyes, because what is the point of having eyes and wasting energy on eyes if there's no light? It's just an absolute waste of energy. So another thing that's very cool about this is you can actually see these two kind of darker spots on this worm, on this polychaete worm, where there used to be eyes. So this is called a vestigial structure. There used to be eyes here, but the worm lives in an area where there's no light. So it's evolved to not waste any more energy on those eyes. So it doesn't have eyes. All right, moving forward. So some adaptations to deal with extreme pressure is to be globby and gelatinous. So this is that sea pig right here that we saw earlier being slurped up by that slurp mechanism. And now we can see that it is clear. It looks like it's mucus. It's globby and gelatinous to deal with that pressure. So if you can't fix it, feature it. Might as well just be super globby and gelatinous to deal with the pressure. And then on the opposite side, we see animals that have extremely hard exteriors to deal with the immense pressure. So this is a deep sea isopod. Okay, some organisms have evolved to have this awesome chemical that is an antifreeze. So these organisms are able to withstand freezing temperatures because they've evolved to have an antifreeze. And humans have seen this and have biomimicked it, meaning we've isolated whatever enzyme or chemical this fish has, and we've synthesized it, we've made it our own, and now we fill our cars with them so that we can drive through Minnesota in the winter months without our pipes and our cars freezing over. So that's a form of biomimicry, where we've mimicked what these fish have created on their own. Evolution. So cool. All right. So in the deep sea, food is few and far between. So these fish don't get to eat meals like humans do. It's not as readily accessible. So they have huge mouths so that they're able to be opportunistic feeders and swallow whatever meal comes their way so they don't miss out on some calories. As seen in this anglerfish right here, that giant mouth, and then also this awesome gulper eel, which is Mbari's emblem. If you see the Mbari logo, you will see this gulper eel on that logo. Okay, so these fish are opportunistic feeders. They have huge mouths to optimize feeding success when there's a chance of catching a prey item.
Yeah, there's something dark over to the left hand side, sort of in the center. It's moving along there. This thing here. Can we take a look at that? What is that?
Oh, wow.
Looks like a Muppet.
What are you?
What is
It's like
that?
Huh.
Oh, it's a
It's
fish.
defensive.
It's in the water column. Is
That's
that
a
what it is?
fish?
What? I don't know.
What?
It
Wow.
looks like
Is it engorged or is it like that?
Is it mad?
Yeah.
Catch
Is
it.
it just?
What? Oh my
God.
God. Wow. Oh my God.
It's kind of like googly eyes.
I think he ate too much.
Yeah.
He's full.
Wow.
Wow.
We really ticked him off.
Uh-oh.
Oh.
What?
Oh, that looks like the gill archer. The gill.
Some kind of,
What is
whoa.
it?
It just got so big.
It's all bladder.
Oh my God.
That's his defense. Like, let me blow up so I can show him how big I am.
Incredible.
Wow.
Yep, he's doing the shake.
What's
Is that a gulper eel then?
Wow.
Yes.
So yeah, that is crazy.
That was awesome.
awesome.
What a way to end the watch, right?
Ah.
So that is one of my all-time favorite videos because you can see the scientists just so giddy. They have an incredible sparkling conversation because they're looking at a gulper eel in real time in real life. So you can see the gulper eel has gulped some seawater and it's very huge, it's robust, and then it slims down to be just a regular gulper eel. So, why does this fish have a small eye, lots of feeling cirri, no webbed fins, a huge mouth, backwards facing teeth, and red color? Think about it for a little bit, pause the video, and then we'll talk about it. Okay, so this gorgeous anglerfish has a super small eye because it's probably actually in the process of losing that eye. What's the point of having lots of energy go towards a organelle or a organ that you don't really need, right? There's no light down here. So what's the point of having an eye do work that's not going to help you out at all? So it has a small eye because it's probably in the process of losing that eye. It has lots of feeling cirri. These are the tendrils that you see protruding from the organism. These are feeling cirri. And the purpose of this is to pick up all of the loss of the sensory that the eyes don't have. So this organism can feel when a fish swims by or a predator swims by using their feeling cirri. It has no webbed fins because this fish is just sitting around waiting for a prey item to swim by. It's not chasing down its prey item. It's hanging out. It's an ambush predator. It's waiting. It's not chasing down its prey. So it doesn't need webbing between its fin rays. It has a huge mouth, so it has a little bit more security in getting that prey item in its mouth, if there is a prey item. It has backwards facing teeth for that same reason. So it really is not wanting, well, evolution. Evolution happened, and it's the purpose of these backwards facing teeth is to grab whatever prey item is there and get it in its mouth. All right, and it has that red color to be cryptic. Red is the first light to dissipate in the upper pelagic. It doesn't make it down to the deep sea, so this fish has evolved to be red, to be camouflaged and cryptic. And this is proof that evolution is the coolest thing ever. Because the deep sea is so weird, bizarre things happen, like no orientation. There's no up or down. So many creatures living in the midnight zone and abyss don't even try to orient themselves. Because what's the point? Creatures swim upside down, vertically. This is a true label your axes moment.
What's that?
Marine snow. Marine snow is poop and carcasses and other organic matter, such as deteriorating rockfish falling down from the upper pelagic through the twilight zone and the midnight zone and falling deeper and deeper. So marine snow is all of this this white, these white dots right here. And this is all organic material. Organic means it has a carbon backbone. It's of biological material. So it's not rocks or something that's abiotic. It's biotic, meaning it's from a living creature. So I was able to go aboard a MBARI research cruise, and I was the only biologist on the cruise. There's a lot of physical oceanographers who were mapping the seafloor, and they gave me the job of taking screenshots whenever I saw something cool. And they had me rate these screenshots from a 1 being kind of cool to a 5, and the 5 being the coolest thing I've ever seen, potential cover of National Geographic. So I had heard about marine snow in school, in the classroom, and then I got to see marine snow in real time and real life, and I got to take screenshots like this. And of course, I was rating them all a 5 because I think they deserve a cover of National Geographic until I saw this. This is a larvation. We are actually related to larvations. They are in the phylum Chordata. They are urochordates. We are vertebrates within Chordata. Larvations are urochordates within Chordata. So they secrete a mucus housing, and a lot of the marine snow actually adsorbs. You can see this picture over here to the right. A lot of this marine snow, this organic matter, carbon-based, adsorbs to the larvation's mucus house. And then because it's adsorbed to that mucus house, when the larvation is done with its housing and it moves on to the next house to filter feed more and do its larvation thing, it ditches this house, swims away to make a new house. All of these organic molecules are adsorbed to this mucus, where this house then falls to the ocean floor, bringing with it all of these organic matter, all of these organic particles. So thanks to larvations, a lot of our atmospheric CO2 carbon is sequestered into the deep sea, where it stays at the ocean floor indefinitely. It also moves a lot of this food from the upper pelagic to the bottom of the ocean, where organisms like sea pigs eat. Okay, so let's finish watching the rest of this video. You can see a giant larvation. There's an ectophid.
Here's a sinker. Especially when you consider like how many.
Okay, so some deep sea environments. The deep sea is not just a sandy bottom that's completely devoid of life. There are diverse environments teeming with life, especially in extreme habitats. There are a ton of species living in these extreme habitats, extreme environments, which I will get into in my next slide. So here is a seamount. This is an underwater mountain that is also teeming with life. You can see some corals right here. These are bubblegum corals and some brachyngeids. And then let's delve into extreme environments. So the first extreme environment that I'm going to talk about are hydrothermal vents. Hydrothermal vents occur at divergent zones and subduction zones on the ocean floor where tectonic plates are moving apart or sliding beneath each other. Water that is geothermally heated underneath the ocean crust comes up through fissures or cracks at up to 700 degrees Fahrenheit. And though water does boil at 212 degrees Fahrenheit, the water exiting the vents is under so much pressure that the water actually does not boil. So, the very first hydrothermal vent was discovered in 1977, just 46 years ago. Actually, it might be 47 now. So the vent was dubbed the name Rose Garden because of the tube worms' alluring, bright red plumes used for respiration. What makes these plumes red, you ask? The answer is blood. Blood containing hemoglobin. And it's called the Rose Garden because of these gorgeous bright red plumes. Again. Methane seeps are a type of cold seep where gas, in this case methane, bubbles or seeps up into the pelagic from the Earth's crust. The main essential difference between hydrothermal vents and cold seeps is that seeps are generally ambient seawater temperature, while vents are super hot, piping hot. Hydrothermal vents and methane seeps are run by chemosynthesis. So these are chemicals that enable primary production and offer a base form of energy. So these right here, these organisms that you see, these beige tubes with the rose plumes, are tube worms. They are types of ecosystem engineers because they engineer an ecosystem for other organisms to live within. So you can see they create a substrate for other organisms. So these tube worms, these vestimentiferan tube worms, thrive off of symbiotic bacteria that actually live inside the tube. And this symbiotic bacteria is able to survive because of these hydrothermal vents and methane seeps. Now let's watch this video.
Look at the other vents we have.
But just go back down, hold out.
Oh, you want to go back down?
Yeah,
Okay.
just go back down, hold out. Just as I'm talking to Chris about trying to do a biomass estimate. That'll give us close and I just get the whole thing here. Yep.
Some of these worms can get as long as 5 feet.
Oh, I think that wouldn't be that hard to do.
Probably my favorite fun fact ever is that the discovery of hydrothermal vents completely changed the way humans defined and interpreted life. So before these vents were discovered, life was thought to need sunlight. So people thought that all life in the world needed sunlight to exist because sunlight, photosynthesis, plants, and then everything else eats those plants. That was a thought process. However, now we know that life can exist in complete darkness thanks to hydrothermal vents and methane seeps. So the fact that life can exist without sunlight has a wonderful application when it comes to finding life outside of our solar system. ... Okay, now for whale falls. So entire ecosystems sustaining off of a single whale that's dead. Once the whale dies, it sinks to the bottom of the ocean, where animals feast on it for years and years. There's a whale off the coast whose name is Rosebud that has been sunken for many years and still has a bunch of organisms living around it. So this is another example of an ecosystem engineer. It's an entire ecosystem that sustains off of a single whale fall. So in this picture, you can see fish, you can see mollusks, octopi, and then you can also see this fuzzy stuff down here in the right hand picture in the bottom of that whale jaw. So that right there is a bunch of worms. They are dubbed the name zombie worm, aka Osidax is their scientific name. And these zombie worms bore into the bones to feed on the lipids within the bone. So here is what they look like from a distance, and then up close, you can see this wonderful shot. These are actually eggs. These are eggs that the zombie worms are pouring out. And this is a female. The male actually lives inside of this zombie worm. It's a parasitic, super tiny male that lives inside the female. Talk about sexual dimorphism. The zombie worm, which is their colloquial common name, aka Osidax, which is their scientific name. So these worms bore into bones to feed on the lipids. They were discovered and described and named by my advisor, Dr. Greg Rouse, in this picture. And here, my advisor is preparing a deployment of an experiment in hopes to collect Osidax worms. So these baskets right here are metal baskets, and we zip tied various types of meat, bones, to the deployment baskets. I think it was chicken bone, beef, and pig bone, along with weights so that they would actually sink. So we deployed them in different areas of the deep sea, and we wrote down coordinates to where these baskets were so that the following year we could actually find them. And we found some awesome Osidax. So, discovering new species in the deep sea. 90% of Earth's habitable space is the deep sea. It's drastically unexplored, which means that there's so many species waiting to be discovered and waiting to be described. There are different estimates that scientists have come up with to estimate how many species are out there waiting to be described. This paper, the first paper published in 2011, estimates about 40,000 to 56,000 species, whereas this next paper right here, called The Magnitude of Global Marine Species Diversity, estimates between 540,000 to 813,000, whereas the bottom paper estimates over 2 million species. So we are still unsure how many species are in the ocean waiting to be discovered. However, I would estimate that there's probably around 2 million species. I agree with the higher estimates on these publications because, like I said, the deep sea harnesses 90% of Earth's habitable space. 90%. That's so much space that animals can live in. And we're just starting to explore the deep sea. So I think that if any of you all want to find new species and name them, you totally can, because there's so many waiting to be discovered. So, what goes into describing a new species? First off, you have to have reason to believe that you have a new to science species. That means you must know your group of organisms very well. Next, you prove that it's new by examining both genetic and morphological data. Morphology is how these animals look. Also structures. So let's say that you pull up this cool looking worm and you're not sure if it's a new species or not, and then you count how many cirri it has. You look at its eye structures and so on and so forth. That's morphology. And then next, you publish your findings, you publish your new description, and then once it's accepted to a scientific journal, then that's that. Your species is a new species, new to science.